All right. Hi, guys. Welcome to week 8. Thanks very much for coming. Just the 3 lectures that we're going to talk about this week. 2 of them are on farm, and one of them is a sort of practical that you may have on the integumentary system. So I'll start with that, and then I'll run you through drugs and neurotransmission, and then hand over to Meg afterwards. So we'll start with the integumentary system, a bit of anatomy for you. And basically, the integument is just like the outer part of the body. So we're thinking just about the skin here and the layers of the skin when we talk about the integumentary system. So what does the skin consist of? Well, we have our epithelium. As with most tissue, we've got our stratified squamous epithelium, so layers of flat cells. Underneath that, we've got our connective tissue, which includes both the dermis and the hypodermis. So the 3 main layers of skin are the epidermis, the dermis, and the hypodermis, also known as subcutaneous tissue, because it's pretty much just fat. This epidermis is what contains the epithelium. The dermis is the main sort of layer of the skin. That's the thickest layer, and that contains most of the structures as you see here. So you might get some muscles called erector pili muscles, which are involved in raising hairs. We have our nerves in here innervating from sensory cells. We've got sweat glands, we've got blood vessels. That's all going to be located in the dermis. So really, we've got like epithelium in the epidermis, major structures in the dermis, and some fatty subcutaneous tissue known as the hypodermis. And as you know, skin can be hairy or non-hairy. Hairy skin is thin, and non-hairy skin, like on your palms, is thick, also called glabrous skin. And this non-hairy skin has a thicker epidermis, so it'll be more layers of these flat cells, as we need that, mostly that's what we're going to be using. So we're going to use that to contact other things, like with your palms, use them all the time. And that will have a more pinkish color, as you can tell by the different pigment of your palm, perhaps to the back of your hand. So we'll start by looking at the epidermis. And while it is pretty much epithelium, there are 5 different layers of epithelium. And we can memorize these layers with the mnemonic, come, let's get sunburnt, because they are CLGSB are the names of the different layers. So starting most superficially, so that's going to be on the outside contacting the actual world, is the stratum corneum, and this consists of dead anucleate, so no nucleus, cells, and some layers of keratin. So that consists of these flat cells at the top. Next is the stratum lucidum, which is again flattened dead cells with keratin proteins, this layer here. Then we have the granulosum, which is our keratinocytes without nuclei. So now we move into more living cells. We have the spinosum, with the keratinocytes connected by desmosomes, which are basically structures that allow cells to be connected together. And we have the stratum basale deepest, connecting the epidermis to dermis. And this is where our keratinocytes and melanocytes will divide and actually form all of these cells. So you might think like basal, I sort of remember it as basement, that's the one that's located the lowest. And that's where we get the cell division. So you can see here, it's actually just a lot thinner than some of the other layers. So key, perhaps when you look at some histological images of skin cells, if you can locate the epidermis, try and find the 5 layers, and then remember this come, let's get sunburnt mnemonic. Okay, and I mentioned before, the dermis is sort of the main action area. That's where most of the major structures are located. And really, the dermis is a layer of connective tissue, and it's separated from this epidermis by a basement membrane. So all epithelium sits on a basement membrane. And the dermis has 2 sort of sublayers. We have the papillary layer, which is loose connective tissue, and that contains lots of cells, has the blood supply to the epidermis, and you can see it here as little fingers or papillae, little protrusions or wave-like shape that protrude into the dermis. So we have some nerves and nerves innervating sensory receptors, and also our blood supply as well, probably some lymphatics too. And under the papillary layer, we have the reticular layer, and that's dense, regular connective tissue with thick bundles of collagen, there for a bit of strength and rigidity. So in the dermis, we have our sensory receptors, which will be innervated by our nerves, cutaneous nerves. And two important sensory receptors are the Meissner's and Pacinian corpuscles. Meissner's corpuscles, you can see one here, sort of a stack of flap cells surrounded by a capsule. So here we have like a bunch of cells all in the same orientation, whereas the Pacinian corpuscle has this sort of circular lamellae shape. Our Meissner's corpuscles are located really superficially, so really like high up in the dermis, very close to the epidermis, and that's because they are the sensory receptor for light touch. So to actually pick up on that light touch, we need them to be closer to the actual surface of the skin. And our Pacinian corpuscles are located deep in the dermis, right? And they're going to sense pressure and vibration. So, you know, it's more of a heavy signal, so that can travel through a bit of skin. So that's why they can be located deep in the dermis. And you'll get lots of these in your fingers, and the nerve fiber is this bit in the middle, surrounded by some layers of tissue. The other structures that you get in the dermis are some sebaceous glands. So these are glands that are often found near hair follicles, and they secrete a milky substance called, an oily lippy substance called sebum. Right, so these cells will lyse and put some sebum onto this hair shaft, and then it'll travel up the shaft and leave the skin, and call this holocrine secretion. These glands become active during puberty, and their aim is to soften and waterproof the skin. We get them in both hairy and non-hairy skin, but particularly in the hairy skin, they will empty into these hair follicles, which allows the contents to more easily reach the surface of the skin. And just with the hair follicles, they are downgrowths of the epidermis into the dermis, right? So the hypodermis is just fat. The base of these hair follicles is in the dermis, and the shaft will be located in the epidermis. Another structure that we'll get, so here is our hair follicle. These are our sweat glands, right? And so there's two different types, as you see here. The first type are eccrine type sweat glands, and they secrete sweat onto the skin surface. So you see with this sweat gland here, it goes straight to the skin. Whereas apocrine type glands secrete their milky substance into the hair follicles. So this sweat gland goes and empties into the actual shaft, and maybe there'll be some sebum as well, and that will come through the hair follicles. So that the major difference is just where the sweat is actually inserted. These glands have their secretory unit, this bundle of fibers in the dermis, and their shaft here is in the epidermis. Some other cells that you get in the dermis and potentially in the skin as well. So adipocytes, they're the fat storing cells found in the hypodermis. They allow it to be that fatty layer. And also in the epidermis, that basal layer, will get our melanocytes. They secrete melanin that gets taken up by the keratinocytes. Melanin protects your DNA, and different skin pigments will produce different levels of melanin, and extra melanin will prevent you from having DNA damage that may lead to cancer. So melanin is good, and that's why different people of different skin pigments have different chances of cancer, because their melanin is at different protective levels. So that's really it for the integumentary system. It's just key to memorize, I guess, or to know the different layers and what structures will be found really in each layer, and try and remember that mnemonic, come let's get sunburned. The next thing I'm going to talk about are drugs and neurotransmission. And really this is quite an important lecture, because it will, you know, set up your knowledge for many future lectures on similar topics. So we start with neurotransmitters. And when we think about nerves, here's just your typical nerve with your dendrites, your cell body, with signals here, eventually creating an action potential that travels down this axon and at the axon terminals. After the axon terminals, there's got to be something else, right? There might be another nerve, so we might have dendrites of a new nerve. We might have muscle here. There's going to be some other structure. But how do we get the message from these axon terminals to the next structure? That's where neurotransmitters come into play. So within a nerve, the signal travels all the way down just purely electrically. But once we get to the end of the nerve, we need substances like neurotransmitters to actually move the signal from one structure to the next. So we have electrical transmission in the nerve and then chemical transmission, maybe from nerve to nerve or from nerve to muscle. So neurotransmitters are substances that allow the transmission of a signal from one nerve to another or a nerve to an effector site. And dysfunction with these neurotransmitters can lead to many diseases, one of which we're going to talk about is myasthenia gravis. So neurotransmitters are normally synthesized and released from the nerve. So the nerve actually makes the neurotransmitters and the nerve releases them, and they can be excitatory or inhibitory. So this nerve gets fired, there's an action potential, it comes all the way down. When we reach the axon terminal, let's say it synapses onto another nerve, this nerve can choose whether it will excite the next nerve or inhibit the next nerve. And that plays an important role. Okay, so there are a few steps in neurotransmission. First of all, we need an actual signal to actually trigger the whole process. So the action potential will travel along the presynaptic neuron. Presynaptic just means like before the synapse. So that's step one. The action potential comes down. Once the action potential sort of reaches this axon terminal, we get opening of our calcium channels. So the local depolarization will cause these calcium channels to open. This opening of these channels will cause our vesicles, which already have our preformed neurotransmitters, remembering that the nerve synthesizes the neurotransmitters and they'll package them in these vesicles. Once the calcium channels open, these vesicles will move towards the end of the nerve and they'll leave the nerve via exocytosis. And so now we have neurotransmitters in the synaptic cleft, and they'll move by diffusion because there's a concentration gradient and cross the synapse in step 4. And step 5, some of these neurotransmitters due to diffusion will bind to receptors on the postsynaptic neuron, maybe one of the dendrites of the postsynaptic neuron, and they will, through signaling, contribute to the next action potential being fired. So you now know the process of how different nerves activate other nerves, and it's a case of actually the message moves via electrical transmission, but the way the nerves activate or inhibit other nerves is by chemical transmission with neurotransmitters. So this is a good diagram to be able to understand, and maybe just have a look at it and see if you can name these 6 steps later on. So when we think about neurotransmitters, it's not just one molecule all the time. There are lots of different neurotransmitters that may be used. And particularly of note are 2 called acetylcholine or ACH and noradrenaline or NA. And these 2 neurotransmitters have particular roles in the sympathetic nervous system and the parasympathetic nervous system, which are 2 systems which control the internal environment in our body. So our sympathetic nervous system, which I'll talk about, will use mostly noradrenaline, whilst the parasympathetic nervous system will use mostly acetylcholine. And so this diagram shows it fairly well. In our somatic nervous system, so if we want to innervate muscle, we have acetylcholine at an N receptor. In the parasympathetic nervous system, we have acetylcholine on the presynaptic nerve. That will bind to an N receptor and go on a postsynaptic nerve, which will also release acetylcholine, which will bind on an M receptor. And I'll talk about the receptors later on. And in the sympathetic nervous system, again, the preganglionic nerve will, or the presynaptic nerve, they do the same thing. They'll release acetylcholine, and then the postsynaptic nerve will more often than not release noradrenaline. Sweat glands are the exception where it releases acetylcholine. Okay, so different neurotransmitters are used in different systems. However, acetylcholine is always used at the ganglia. So from nerve to nerve, acetylcholine is always the neurotransmitter. And the only time we actually see noradrenaline here is at postganglionic sympathetic synapses. So here, we have a preganglionic, which releases acetylcholine, and a postganglionic nerve, which releases noradrenaline. The only exception to that is at sweat glands, where your sympathetic, postganglionic sympathetic nerve will release acetylcholine. Okay, and these nerves that release noradrenaline are called adrenergic nerves, and the nerves that release acetylcholine are called cholinergic nerves. So cholinergic nerves, they will release acetylcholine, and that acetylcholine will bind to either a nicotinic N or a muscarinic M receptor. N receptors are activated by nicotine, as the name suggests, and we get them at the ganglia. So between presynaptic and postsynaptic nerves, we find them in the brain, and we also find them at the skeletal neuromuscular junction. So your muscles have N receptors. M receptors, however, are activated by muscarine, which is a poisonous mushroom, and they're found at parasympathetic organs and the sweat glands, which is the exception to the sympathetic nervous system, remember. So the way I generally remember it is like parasympathetic system is M receptors, and the sympathetic system is, as we'll talk about, alpha or beta receptors, and N receptors are everywhere else. So like your neuromuscular junction, the ganglia, and in the brain. And you just need to remember that sweat glands are the exception. So acetylcholine, which is a pretty key neurotransmitter, as you see, it has impacts somatic, sympathetic, and parasympathetic nervous system just because it's always at the ganglia. That's formed using the enzyme choline acetyltransferase, and it's inactivated, broken down by the enzyme acetylcholinesterase, and it's inactivated in the synapse. Now, some conditions will have overactive acetylcholinesterase, and obviously if that happens, that'll impact the ability of acetylcholine to actually bind and activate these nerves. So we might have some drugs that target that. And our N receptors are ion coupled or ionotropic, meaning when the neurotransmitter binds, our ions can come through, whereas our M receptors are G protein coupled or metabotropic, meaning our neurotransmitter binds, that will activate a G protein, which in turn activates an effector protein, which in turn impacts the ion channel. So don't really need to know how it works, just that there are a couple of types of ways the ions actually move into the cell, and the two receptors are two different types. Now with adrenergic nerves, there are a few neurotransmitters that they'll release. Noradrenaline is the main one, but they can also release adrenaline or dopamine. And these neurotransmitters act on alpha receptors, or the beta receptors. And both of these are metabotropic, so G protein coupled. Noradrenaline is synthesized from tyrosine, and it is removed from the synapse by two pathways. The first of which is called uptake 1, which involves the noradrenaline being uptaken into the first nerve or the presynaptic nerve. And the second pathway is uptake 2, where the neurotransmitter is taken into the second nerve or the postsynaptic nerve. Both these pathways are active, so they require energy. And once in the nerves, we have two enzymes, MOA and COMT, which will break down the enzymes. So adrenergic nerves, they innervate lots of different structures. You see here the eye, heart, lungs, blood vessels, gastrointestinal tract, bladder, and sweat glands. These are just any internal structure pretty much which is going to be impacted by the sympathetic nervous system. And cholinergic nerves from the parasympathetic system will produce the opposite impact in pretty much all of these structures, right? They also, cholinergic nerves also will act on skeletal muscle. So it can be helpful to know some of these receptors, and really the response is just the opposite. This is the sympathetic table. The response is quite simply the opposite if you're looking at a parasympathetic response. So for example, your sympathetic response, your fight or flight, that will involve increasing your heart rate. If you saw a serial killer at your door, your heart rate would go up, and your lungs would dilate to get some more air in, your pupils would dilate so you could see what's going on. Your blood to certain areas would vasoconstrict, minimizing blood flow to places like your gastrointestinal tract because you don't need to be digesting things, whereas your blood flow to skeletal muscle would increase because you might need to move and run away. As mentioned, you don't need to be digesting, so your gastrointestinal motility will decrease. Some of your sphincters will constrict, again inhibiting digestion, and your sweat glands will be stimulated. So I think that the main sort of receptors here, if you're looking at how you remember them, a good one is we have and again there's subtypes of the A and B receptors. Really, I think the one that's important to know is we get B1 receptors in the heart and B2 receptors in the lungs. And you can remember that because we have one heart, so B1, and we have two lungs, so B2. The other thing just generally is that activation of alpha receptors will constrict blood vessels, and activation of beta receptors will dilate. And the way I remember that is like A comes before B in the alphabet, and vasoconstriction comes before vasodilation as well. Like constriction is alphabetically ahead of dilation. With the parasympathetic nervous system, it's a little bit more straightforward. It's just M2 in the heart and M3 pretty much everywhere else. Okay, so what happens at the neuromuscular junction, or the NMJ? So this is when our nerve actually targets the muscle. So we have a little gap here, and that the signal crosses, and that gap is termed the motor end plate. And the transmission will occur via acetylcholine, which is these little mouses here, and that will act on N receptors, and activation of those will open our sodium channels and cause the depolarization of this muscle. So we can have drugs which act presynaptically, so on this actual nerve to block the release of acetylcholine, or we can have drugs that act postsynaptically, so on the actual receptor site to inhibit the binding of acetylcholine to these sites. And here are examples of these drugs. So it's important to realize that for acetylcholine to actually work as a neurotransmitter, it has to be removed almost immediately. So it has to activate and then stop activating it. So there's two ways we can, if we don't want this effect, there's two ways we can block acetylcholine from working. The first is constant agonist stimulation. And if you can remember, an agonist is like when the key fits the lock and opens the lock. So these actually activate the receptors. And agonists in this case are called depolarizing blockers because they actually depolarize the muscle, as it may be. The catch is they keep the action potentiated. So they constantly activate the receptor. And that is pretty much the same as if you block the receptor, because remember, you need to remove acetylcholine almost immediately to actually have an effect. So if you just keep the key and the lock open the whole time, there's no effect. And that will mean your N receptor is, and the ions become unresponsive to acetylcholine. And an example of a depolarizing blocker is succinylcholine, but we don't use that much. So constant stimulation with an agonist prevents the cycles of depolarization and repolarization, which are required for sustained muscle contraction and relaxation. So if you constantly activate the receptor, it pretty much doesn't work. The opposite to that, but again, produces the same effect, are non-depolarizing blockers. So these are antagonists. This is like a key that fits the lock but doesn't open the lock. So they bind to the receptor, but they don't activate it. And if they bind to the receptor, it means acetylcholine can't bind to the receptor, so we don't get that effect. We can overcome the action of these non-depolarizing blockers by just increasing the concentration of acetylcholine, because then it'll beat the drug to the receptor. And examples of these are tubocararine, which is an older one, and rocuronium, which is a newer one. Why would we use any of these? Well, we might want to produce paralysis, temporary, of course, maybe before surgery, something like that. If the patient is unable to breathe, that's probably due to smooth muscle constriction in the airway, so we want to relax that. And yeah, these two methods will produce muscle relaxation. So we can't produce the signals that actually contract the muscle, because there's some sort of blockage going on between the nerve and the muscle. And this brings me to the condition I talked about before, myasthenia gravis. This is an autoimmune disease which affects neuromuscular transmission. And basically, we have a reduced number of N receptors at the motor end plate. So the motor end plate being this sort of area here with all the receptors. So if the body attacks some of these N receptors, then we have less N receptors, and we have so little that, even if we activate them, we don't get enough signal to have a full or sustained muscle contraction. So this condition, which involves autoimmune attacking of these N receptors, will result in muscle weakness after activity, but you'll recover after a short bit of rest. So you won't get that weakness. But again, you can't put too much strain on your muscles because their actual ability to fully contract is impaired, as we see here. So how do you treat it? Well, we treat it with anticholinesterases. Remember, cholinesterases break down acetylcholine. So anticholinesterases will inhibit that breakdown, and that will increase the concentration of acetylcholine in the synapse by preventing those activity of those enzymes. And an example of an anticholinesterase is pyridostigmine, and that's reversible. We can also use competitive antagonists at M receptors. So remember, this is acetylcholine acting on N receptors, but acetylcholine could also act on M receptors, and we don't want the parasympathetic response all the time, right? Because if we have prolonged parasympathetic activation, it's not good for the body. We need balance. So if we are going to increase the concentration of acetylcholine, we don't want acetylcholine binding to M receptors. And so we have a competitive antagonist, so a drug that will compete with acetylcholine to bind at this M receptors, but binding won't do anything. And that will just be used to block unwanted parasympathetic effects. And an example of a competitive antagonist at M receptors is atropine. So yeah, I think this is a good condition to know. And I think you might get asked something about myasthenia gravis, or perhaps something about some of these drugs, which is perhaps a little bit unfair, but there's not too many of them. I'll now hand over to Meg to talk about anti-cancer drugs.
Right, so there's quite a few different drug names to go through in this one, but as long as you kind of get a brief overview of everything, that's what we're looking for. So the learning outcomes outline the principles of cancer therapy, identify targets for anti-cancer drugs, describe the mechanism of action of classic cytotoxic drugs, discuss the major adverse effects of anti-cancer drug therapy, discuss strategies for the management of adverse effects, describe the mechanism of action of novel anti-cancer drugs, and describe common clinical approaches to drug therapy and the limitations of cancer chemotherapy. So there's kind of 4 different main areas of cancer treatment. So you've got chemotherapy, which I'm sure you've heard about before, surgery, radiotherapy, and then supportive therapy. So that basically covers anything that's not the above 3. And the main things we're going to go into in this lecture are chemotherapy and supportive therapy. So starting off with chemotherapy. So we're going to talk about the principle of selective toxicity to start off with. The primary aim of cancer drugs is to have selective toxicity, meaning they will specifically target cancer cells. Cancer cells can be identified by unique structures or uniquely expressed elements which normal cells wouldn't have on them. However, adverse effects do still occur even when you've got this principle of selective toxicity, particularly in rapidly dividing cell populations. And this is because cancer drugs specifically target cell division, because I'm not sure if you've done much biochem around cancer yet, but one of the key things about cancer cells is that they rapidly divide. So cancer drugs are made to specifically target this rapid division, but this also has side effects, which include your hair falling out, because your hair, like the cells that produce hair, that's a rapid turnover. It can lead to infertility in your sex cells, skin blisters, because skin cells are also rapidly dividing, GIT ulcers and inflammation, a decreased red blood cell, white blood cell, and platelet count, dysrhythmia or altered contractility of the heart, kidney failure, damage to microtubules in the neurons, or bladder bleeding and inflammation. So for different types of drugs, we've got the classic cytotoxic agents. So these are kind of the older chemotherapy drugs. These drugs essentially damage all of your cells, hence the name cytotoxic. And generally they interfere with the process of DNA transcription and translation. So if you cast your mind back to the DNA lectures from the first 2 weeks, we've got alkylating agents, antimetabolites, plant alkaloids, and hormonal agents and antagonists. Then we've got novel anti-cancer agents. So these are some of the newer types of drugs. Some of these are probably still in their trial phases, and these work slightly differently. So we've got monoclonal antibodies and immunomodulating agents. So for the classic cytotoxic agents, alkylating agents. So alkylating agents insert an alkyl group into DNA, which forms intrastrand and cross-links between bases on the same and opposite strands. This is especially between guanine bases. This stress causes the DNA strands to break, which induces apoptosis. And these are cycle non-specific. There are many side effects associated with the use of alkylating agents, as they can affect many different cell types. And one of the drug examples for alkylating agents is cyclophosphamide. Then we've got cytotoxic antibiotics. So these are antibiotics that inhibit transcription and translation of DNA. However, they're very toxic, so you wouldn't use them as normal antibiotics when you've got some sort of infection. They're only used in cancer patients where the benefits of fighting the cancer outweighs the risks and the toxic side effects of these drugs. So what they do is they can donate electrons, which result in the production of highly reactive oxygen compounds, or superoxide, and this causes breakage of the DNA strands. This prevents the religation of DNA. The side effects of cytotoxic antibiotics include cardiotoxicity, nausea, vomiting, and hair loss. Cytotoxic antibiotics affect all cells with a fast turnover rate. And an example of a cytotoxic antibiotic is doxorubicin. Then we've got antimetabolites. So these drugs substitute themselves into the folate pathway during the synthesis of DNA, and they are cycle specific, meaning they only target one portion of the mitotic cycle. The side effects include affecting all cells with a fast turnover rate, photosensitivity, nausea, vomiting, and ulcers, and they cannot be given to pregnant women. An example is methotrexate, which is a competitive inhibitor of dihydrofolate reductase. That's just an enzyme in the folic acid pathway, which is part of the nucleotide synthesis pathway. Then we've got histone deacetylase inhibitors. So these just prevent the removal of acetyl groups in the histone complex. This affects gene expression and the progression of the cell cycle, which causes the cells to trigger apoptosis. Plant alkaloids. So these are another type of drug that is cycle specific. And we've got 2 different ones. We've got mitotic poisons. These inhibit microtubule function, which stops the spindle from forming during the M phase of the cell cycle. And then we've got topoisomerase inhibitors, which disrupt the coiling and uncoiling of the DNA. So this stops S phase from occurring. Now we're moving on to hormones and hormone antagonists. So we've got selective estrogen receptor modulators. So these drugs act as antagonists to suppress cancer growth with anti-estrogenic action for breast tissue. By specifically targeting the estrogen receptors in the breast, osteoporosis can be avoided because estrogen is also important in your overall bone health. Side effects include hot flushes and sweating, and one drug example for this is tamoxifen. Next, we've got aromatase inhibitors. So aromatase inhibitors act on aromatase, which is an enzyme in estrogen synthesis. By inhibiting aromatase, the circulating levels of estrogen decrease. Side effects are mainly related to the GI system, and an example of this is anastrozole. Then we've got gonadotropin-releasing hormone analogues. So gonadotropin-releasing hormone analogues synthetically stimulate the release of androgens. The repeated release of androgens produces negative feedback, which reduces the amount of androgens in circulation, and this is used to treat prostate cancer. Got glucocorticoids. So these are used in high doses to induce lysis of affected cancer cells. These are used to treat blood-borne cancers such as leukemia and lymphoma. So those were all of the main different categories of drugs that we have for chemotherapy. And I know I just threw a lot of information at you, but the most important part is to learn the different methods of actions for those drugs instead of learning the specific drug names. Obviously the drug names are like helpful, but I would say it's better to understand the principles of how they work. So now onto other supportive therapies. So these aren't chemotherapy drugs, but these are just other measures that you might include in a cancer treatment plan. So we've got antiemetic drugs. One of the major side effects of chemotherapy is emesis, which is just another word for vomiting. But there are many different antiemetic drugs, which include dopamine receptor antagonists. One example is metoclopramide. We've got serotonin receptor antagonists, such as ondansetron, glucocorticoids, such as dexamethasone. Then we've got colony stimulating factors. So this is another type of supportive treatment, which basically helps your body produce more blood cells. So erythropoietin stimulates the red blood cells and the white blood cell count. And then we've got filgrastim, which stimulates neutrophil production. Monoclonal antibodies. So monoclonal antibodies are a new form of cancer treatment. They're one of the more recent inventions. So this type of treatment involves producing antibody-forming cells in mice, which are then extracted and fused with cultivated tumor cells. So essentially you get this hybrid cell that's called a hybridoma, where you've got an antibody-forming cell mixed with a tumor cell. And the good thing about a tumor cell, well, it's not good when it's in cancer, but because they rapidly proliferate, you'll get a lot of these hybridomas formed in a short period of time. The hybridomas are then screened, and only the ones that are successfully merged and that can produce antibodies, those ones are selected and cloned to make more of the successful ones. The monoclonal antibodies, which are produced by the hybridomas, can be isolated, so like taken out of the cells. And then essentially you've just got lots of these monoclonal antibodies. So one example is trastuzumab. This binds to human epidermal growth factor 2, HER2, and this is overexpressed in certain forms of breast cancer. So if you have these monoclonal antibodies, which are specifically made to target HER2, then they can bind to these breast cancer cells. This helps like label them or mark them out so that then the classic cytotoxic agents, which we were talking about before, they can specifically target these cells so they know which ones to attack. And this kind of helps reduce side effects because it means they're not targeting all of your body cells, but only the cancer ones. Obviously, there are still some side effects. So we've got GI and cardiac effects. And these types of drugs are also expensive because obviously there's quite a long process to produce them. I believe that's it. We've got one more, the biological response modifiers. So interferons augment the cytotoxicity of immune cells, inhibit cell proliferation, facilitate cytotoxicity, and alter antigen expression on tumor and immune cells. Then we've got interleukin-2. This stimulates lymphocyte proliferation and improves cellular immunity, and cytokine treatment, which involves removing a sample of patient's own lymphocytes, growing these in a culture and sensitizing these against tumor cell antigens. This then results in a more aggressive immune response, so the body's own immune response towards tumors. Protein kinase inhibitors. So these inhibit protein kinases, which are essentially signaling molecules essential for some proliferation and activation pathways. So this inhibition can restrict some leukemia and GI tumors and inhibit angiogenesis, which is the creation of new blood vessels to form tumors. We've got intracellular signaling inhibitors, which target molecules such as the RAS protein and cyclin-dependent kinases, and then sensitizing agents, which involve injecting vaccines such as BCG into tumors, which induces an immune reaction in these cells towards the antigens. This then induces an inflammatory response, and some superficial cancer cells are likely to be killed by the immune cells in the process. And then we've got treatment complications. So one thing that happens in chemo is that you develop drug resistance. So tumors can develop mutations which cause adaptations or just varying versions of the cancer cells. And this protects tumors against certain drugs which were previously effective. So that's basically the same principle as natural selection, but on tumors instead. So it means that your tumors adapt to kind of survive the different chemo drugs. Additionally, cancer cells can grow in inaccessible compartments in the bodies or tumor cell sanctuaries. So this is essentially, they grow in locations where the chemo drugs can't reach them. So that's why it's called a sanctuary. And that creates a site of relapse where the cancer is free to grow. Or we've got dose exhaustion. This is where the maximum non-toxic dose is not sufficient to attack the cancer. So the non-toxic dose is the amount that your body can handle without the side effects being so great that it would be too harmful to use. So if you get up to that threshold and the cancer's still there, then that's dose exhaustion. However, there are some strategies to combat drug resistance. So we've got multiple cytotoxic drug therapy. That's where you use lots of different drugs with differing actions at the same time. So this means that you can avoid dose exhaustion because you're using different drugs, so you can use lower doses. And this kills more cells because even those that are immune to one drug likely won't be immune to a different one. We've also got a combination of radiotherapy, chemotherapy, and surgery. So surgery or radiotherapy might be able to reach drugs that are in tumor cell sanctuaries, reach cancer cells that are in these sanctuaries where drugs can't. And this can also overcome dose exhaustion. And then we've got a drug holiday. So this essentially helps prevent the risk of the development of resistance because you just stop medication for a period altogether. And then in this break, it's not exactly known why, but cancer cells will stop dividing sometimes. The effectiveness of drugs are then improved, and some cytotoxic activity does still occur with the lyased cancer cells, killing some of their surrounding cancer cells. The aftermath effects of the drugs can still be active during these break or holiday periods. Cool. So that was it for this week's lectures. Hopefully that all made sense. Thanks for coming along even on a public holiday at the end of mid-sem break. Hopefully that was helpful, and we look forward to seeing you next week.